Hello. For the class of Genetic Engineering project, as for the Exponentias project, we will talk about the use of the expression of endolysins from bacteriophages to counteract the antibiotic resistance of Clavibacter michiganensis.
So, one of the main problems in the use of antibiotics in agriculture is that their misuse can promote resistance to pathogens present in the plant, and that resistance can disseminate through horizontal gene transfer between bacterial cells.
In the agronomical sector, several issues have been found regarding these resistant bacteria in crops, particularly in San Luis Potosí, Mexico. In the tomato farms, there's a bacteria called Clavibacter michiganensis that is responsible for the generation of the canker disease in tomatoes, which leads to a poor harvest and therefore an economical loss for the producers or the farmers.
And now onto bacteriophages. These are viruses that can infect certain bacterial species, and during the last stage of phage infection in both lysogenic and lytic pathways, the bacteriophages produce endolysins, which are enzymes that can degrade the peptidoglycans in the cell's periplasm and consequently lysin the cell for the release of the viral replicates.
In nature, bacteriophages CMP1 and CN77 infect and destroy C. michiganensis by producing some lytic enzymes called endolysins. Cloning these pest-specific enzymes into a plant transforming system will confer resistance to the infection to the resulting plant without the need of antibiotics.
The methodology involves, firstly, endolysins PCR. After that, and with the appropriate restriction enzymes, it has to be carried out the construction of the recombinant vector. And eventually, it has to be created a recombinant E. coli with the novel lytic genes, since these properties have been reported to be successfully expressed in this bacteria. After the harvest of E. coli, the plasmid DNA should be extracted and then introduced into the agrobacterium cells to use it as a vector to insert the genes into the tomatoes. And in order to verify the lytic activity, tissue culture and SES page had to be monitored.
Our business's value proposition offers a C. michiganensis free and resistant plant, which offers risk prevention of the crop losses and also reduces the use of agrochemicals such as antibiotics. The target market will be the important tomato producers in the country and some historically affected zones such as San Luis Potosí.
Some of the impacts of this project are that the costs of the maintenance and preventive measures against the dissemination of this bacteria will decrease. Also, there will be a significant reduction in the use of antibiotics since the plant will be able to produce the enzymes that lyse the bacterial cells. Additionally, there will be a decrease in losses, which will increase the quality of production. And finally, the technology could be implemented on other species.
Here you can find more details of the project and its references.